Hello everyone. Today we are going to study about chemical mediators of inflammation. Now before starting today's topic, we'll summarize the previous topics of this chapter. In this chapter we have studied about the basic things of inflammation, its definition, signs and its types, and the two types of events that occur during acute inflammation. They are of two types, vascular events and cellular events. Vascular events also include two processes, hemodynamic changes and altered vascular permeability, and cellular events also include two types of processes, exudation of leukocytes and phagocytosis. In our previous video, we have completed the cellular events. So today we are going to continue with chemical mediators of inflammation. The mediators are nothing but the substances, the chemical substances which initiates the process of inflammation. So they are also called as permeability factors or endogenous mediators which increase the vascular permeability. If you remember in yesterday's topic, in case of altered vascular permeability, there are 5 mechanisms, and in that mechanism we have studied that there are some substances secreted which are responsible for those mechanisms, for the change in vascular permeability, and that is those are the chemical mediators. So they change the vascular permeability, that's why they are also known as permeability factors. And endogenous mediators, because these substances are synthesized in our body, within our body, due to the as a reflux process to the injury. So that's why they are endogenous substances, they are synthesized within the body, right? So they are either also known as endogenous mediators or also as a permeability factors which enhances the vascular permeability. However, many chemical mediators also take part in other processes of acute inflammation as well, like vasodilatation, chemotaxis, fever, pain and tissue damage. We have also seen that in vascular events and cellular events that vasodilatation, increase in blood vessels diameter, then chemotaxis is the attraction of WBCs from the blood vessel to the tissue, interstitial tissue is called as chemotaxis. So those things, those mechanisms are also responsible, they are also produced due to the chemical mediators, the secretion of chemical mediators, right? So acute inflammation, all the mechanisms are somewhat connected with the secretion of these chemical mediators within the body, right? Now mediators may be released from the cells, plasma or the damaged tissue itself. So based on that, they are classified in two groups. First, mediators released from cells and mediators originating from plasma. So based on their origin, based on their secretion or synthesis, they are divided into two groups. Those they are synthesized from cells and those they are originated from plasma. So there are two types of mediators, starting with first one, cell derived mediators. So those mediators synthesized from cells include, first one, vasoactive amines. There are two vasoactive amines, histamine and 5-hydroxytryptamine, which is in short also known as 5-HT. And also the synonym of 5-HT is serotonin. Serotonin. So there are two vasoactive amines, histamine and serotonin, or you can say 5-HT. The first, histamine, it is stored in granules of mast cells, basophils, platelets, and they are released from cell by various agents like stimuli or substances inducing acute inflammation, that is cold, heat, radiation, immunological reactions, etc. histamine releasing factors from neutrophils, monocytes and platelets, and interleukins. So it means histamine where it is synthesized. So the major part of the histamine is synthesized in mast cells, the granules of mast cells, as well as the other cells include basophils and platelets. So this majorly three cells, mast cells, basophils and platelets, secretes histamine, and that secretion of histamine is based on another three things. ક્યારે histamine નું secretion થશે તો કે આ ત્રણમાંથી કોઈ બી condition present હશે ત્યારે એનું secretion થશે. Which conditions? First condition, any stimuli or any substance which induces the acute inflammation. તો આપણે સ્ટાર્ટિંગમાં જ જોયા હતા કે there are some agents, chemical agents, physical agents, immunological agents which are responsible for the inflammation. So any of that agent present in that cold, heat, radiation, like physical agents, any chemical agents like acids, alkali, or poisonous substance, or immunological reactions, any. So or you can say any microbes also, infectious agents. So whenever any injurious agent is present in the body, then these mast cells, basophils and platelets secrete histamine. That is the one condition. Second condition, histamine releasing factors from neutrophils, monocytes and platelets. There are some particular substances too which stimulate the secretion of histamine. So those substances or those chemicals are known as histamine releasing factors. Those factors which stimulate the histamine release are called as histamine releasing factors, and those substances are secreted from neutrophils, monocytes and platelets. So any particular agents, any injurious agents when entered into the body, then they first can secrete histamine releasing factor from these cells, neutrophils, monocytes and platelets, and then those factors stimulates the histamine secretion. So that is somewhat indirect mechanism. First there is release of histamine releasing factors and then it secretes histamine. Okay, and third mechanism is interleukins. Interleukins are another chemical mediators which are secreted in response to inflammation. We will study the later on, right? So whenever there are three conditions, any stimuli which directly stimulates mast cells, basophils, platelets and release histamine. Second, any stimuli or substance which release first histamine releasing factor and then indirectly stimulates histamine secretion. And third is the presence of interleukins. So these three conditions leads to the secretion of histamine. And what the histamine does, it produces vasodilatation, increase in vascular permeability, itching and pain. Histamine is responsible for four symptoms, four mechanisms of acute inflammation. Which four? First, vasodilatation, the dilatation of blood vessels, increase in vascular permeability. If you remember the mechanism, in case of first mechanism, contraction of endothelial cells, this histamine produces the increased vascular permeability. So by contraction of endothelial cells, it increases vascular permeability, itching and pain. Itching, that means that irritation, ખંજવાળ આવે at the site of inflammation. જે જગ્યા inflammation થતું હોય તે તમને itching થાય, right? So these four mechanisms, four responses are produced due to histamine. Okay. Second, 5-HT, or you can say serotonin. The serotonin is present in tissues like chromaffin cells of GIT, spleen, nervous tissue, mast cells and platelets. So they are also secreted from 5 tissues: the chromaffin cells of GIT, spleen, nervous tissue, mast cells and platelets. You can see mast cells and platelets are the two common cells for these both vasoactive amines. So you can also say that mast cells and platelets both are responsible for the secretion of both vasoactive amines, histamine and 5-HT. Right. Okay, now the actions. Actions are similar to histamine. That means vasodilatation, increased vascular permeability, itching, pain. All those four actions are same for 5-HT, but they are less potent than histamine. So that they are less severe than histamine. Okay, so both the vasoactive amines produces same action, in which histamine produces more potent action while 5-HT produces less potent action. Clear? So that is about vasoactive amines. You should include the three things in both the cases: which cells secrete this substance, second, which are the agents responsible for their secretion, and what are the actions produced by this substance. Okay. Now the second one. The second cell derived mediators are arachidonic acid metabolites, or you can say eicosanoids. Eicosanoids. The arachidonic acid is a fatty acid which is also called as eicosanoic acid, and its sources are leukocytes, platelets, mast cells, endothelial cells are the major source. They are also produced from diet directly because it is a fatty acid. So it is also produced from diet. Conversion of essential fatty acid, linoleic acid, to the arachidonic acid. There is one reaction produced in the body, that conversion of the linoleic acid, another fatty acid, into arachidonic acid. So arachidonic acid is a fatty acid which is produced from three things. One, from the cells like leukocytes, platelets, mast cells and endothelial cells, produced directly from diet. And third, conversion of linoleic acid to arachidonic acid. Right. Arachidonic acid, the synonym is eicosanoic acid. So whenever because it is called as eicosanoic acid, the metabolites of arachidonic acid are known as eicosanoids. The metabolites of eicosanoic acid, eicosanoids. Right. Now arachidonic acid must be first activated by the stimuli or the mediator like C5A to form arachidonic acid metabolites. So in this pathway, to produce eicosanoids, to produce the metabolites from arachidonic acid, the first step must occur is the activation of that fatty acid, activation of arachidonic acid, and that activation occur by the stimuli. That means any agent જે આપણે સ્ટાર્ટિંગમાં જ કીધા કે physical, chemical, injurious, કોઈ બી agent, whenever the injurious agent is present, they can directly stimulate arachidonic acid, or some substances like C5A. What is this C5A? If you remember, in yesterday's video, there is C3 and C1. That C means complement system. So C is nothing but the proteins of complement system. So it is the fifth 5A protein, complement protein of the complement system. So it is a complement protein 5A. So whenever any injurious agent which secretes complement protein C5A, so that leads to secretion or synthesis of arachidonic acid by activating the fatty acid. So that is indirect mechanism, right? So when the first step is the stimulation of arachidonic acid, and then that stimulated arachidonic acid is then converted into eicosanoids by two pathways. First pathway is called as cyclooxygenase pathway, and second pathway is known as lipoxygenase pathway. Okay, so first pathway is cyclooxygenase pathway, in which three eicosanoids can be produced, three types of eicosanoids can be produced: prostaglandins, thromboxane A2 and prostacyclin, right? So prostaglandin is we will write now in short PG. So that PG means prostaglandin. So prostaglandin and their related compounds are also called as autocoids. એનું એક સિનોનિમ બી છે ઇકોસાનોઇડ્સ માટે. They are also known as autocoids. Okay. Now the cyclooxygenase pathway. Cyclooxygenase is nothing but one enzyme. It is a fatty acid enzyme which acts on activated arachidonic acid to form prostaglandin G2. So first of all, what happens in cyclooxygenase pathway? You can see in the flow chart. See, activated arachidonic acid by cyclooxygenase is converted into first PGG2. PGG2 is a prostaglandin G2. Now what happens? That PGG2 is enzymatically transformed into PGH2 with the generation of free radicals of oxygen. So PGG2 by another enzymes is converted into PGH2, that is prostaglandin H2 and free oxygen radicals. Okay. Now this PGH2 is further acted upon by enzymes. The another enzymes converts that PGH2 into three types of metabolites. That is, you can see in the flow chart, PGD2, PGE2, that is prostaglandin D2 and E2, prostaglandin F2 alpha, then thromboxane A2 and prostaglandin I2. And this prostaglandin I2, PGI2, is known as prostacyclin. So you can say that PGH2 is converted into three types of substance: prostaglandins, which includes three prostaglandins, D2, E2 and F2 alpha, then thromboxane A2 and prostacyclin. So, and there are different roles of these substances for the acute inflammation. You can see that PGD2 and E2 produces two things: vasodilator, it is vasodilator, and bronchodilator. So it produces dilatation of bronchioles, lungs and blood vessels. PGF2 alpha is a vasodilator, but it is a bronchoconstrictor. It causes the constriction of bronchioles. Thromboxane A2 is a completely reverse. It is all vasoconstrictor as well as bronchoconstrictor. And the third also, there is a one mechanism, platelet aggregation. It induces the platelet aggregation. If you remember the anatomy and physiology, whenever there is a platelet aggregation, platelet form aggregates, they get activated and activates the clotting factors, and clotting factors activation leads to clotting. So whenever there is a mechanism of platelet aggregation, they induce blood clotting. So whenever there is a secretion of thromboxane A2, it stimulates clotting, while the prostacyclin is completely opposite to thromboxane A2. You can see it is vasodilator, bronchodilator and anti-aggregating agent. Platelet aggregation is also inhibited, so it inhibits blood clotting. So prostacyclin is vasodilator, bronchodilator and anti-aggregating agent. Okay. So that is the cyclooxygenase pathway. Okay. Now the second pathway is lipoxygenase pathway. Lipoxygenase pathway produces two types of substance, 5-HETE and leukotrienes. In case of this pathway, the enzyme lipoxygenase acts on activated arachidonic acid to form hydroxyperoxy compound, which is known as 5-HPETE. The full form of that is hydroxyperoxy eicosA tetraenoic acid. Right, tetraenoic acid. That's why it is TE. Okay, so the eicosahexaenoic acid is the synonym of arachidonic acid. So it is converted to hydroxyperoxy compound of arachidonic acid, which is 5-HPETE. Okay. Now that 5-HPETE further undergoes the peroxidation to forms two types of substance. One, 5-HETE, that is only hydroxy compound of eicosahexaenoic acid, which is responsible for the chemotaxis for neutrophils. Chemotaxis શું હતું ફરીથી? Summarize, please. Summarize. Yeah, the chemotaxis is the attraction of neutrophils, or you can say WBCs, from the blood vessel into the interstitial tissue. Right. So these 5-HETE, when synthesized in the in injurious part, that local site, it causes the attraction of neutrophils at that site. So the neutrophils transferred from blood vessels into the damaged tissue. Right. So that is 5-HETE, one mediator. And second, that 5-HPETE is converted into LTA4. LT is the leukotrienes. Leukotrienes are nothing but the slow reacting substances of anaphylaxis. Anaphylaxis એક ટાઈપનું allergic reactions છે. તો anaphylaxis means allergic reaction induce કરતું એક substance which is a slow reacting substance are the leukotrienes, and they are LTA4, LTB4, C4, D4, E4. 5 types are there. So LTA4 is first synthesized, but it is unstable. So it is then converted into B4, C4, D4 and E4. From that, LTB4 is responsible for the chemotaxis for phagocytic cells. So phagocytic cells ma kaya avse? Phagocytic cells, if you remember, we have included that in phagocytosis. There are two types of phagocytic cells: neutrophils and macrophages. Remember? Yeah, macrophages. So that neutrophils and macrophages both are attracted, or you can say transferred, from the blood vessels to the injurious tissue or damaged tissue. So that is the LTB4. LTC4, D4 and E4, all these three has a common functions like smooth muscle contraction. They produces contraction of smooth muscles, hence produce vasoconstriction, bronchoconstriction and increase vascular permeability. So because of the smooth muscle contraction, they produces blood vessel contraction and bronchial constriction and increases the vascular permeability. Okay, so the second cell derived mediators are arachidonic acid metabolites. What you should include in that? First, what is arachidonic acid? It is a fatty acid. The synonym of that is eicosahexaenoic acid. Then it is derived from 3 places. Either they are synthesized in the body. Second, they are obtained from diet. And third, they are converted from linoleic acid to arachidonic acid. Right. Then the main thing, arachidonic acid is first activated by the stimuli or the mediators like complement protein 5A, and that activated arachidonic acid then converted into another mediators by 2 pathways, that cyclooxygenase pathway and lipoxygenase pathway. Cyclooxygenase pathway produces 3 types of substances: prostaglandins, thromboxane A2 and prostacyclin, while lipoxygenase pathway produces 2 types of substances, that is 5-HETE and leukotrienes, LTB4, C4, D4 and E4. Okay, so that is the second mediator. Now the third mediator is lysosomal components. The inflammatory cells, neutrophils and monocyte contain lysosomal granules, which on release elaborate a variety of mediators of inflammation. So lysosomal components are nothing but the lysosomal granules which secretes the mediators, the lysosomal components. And they are mostly secreted from two types of cells, neutrophils and monocytes. Okay. Now, these are the granules, first of all, granules of neutrophils, those substances released from neutrophils. There are two types of granules, specific or secondary and eosinophil or primary. Specific or secondary contain lysozyme, alkaline phosphatase and collagenase. That include three types of enzymes: lysozyme, alkaline phosphatase and collagenase. And eosinophil or a primary have also three types of enzymes: MPO, MPO is myeloperoxidase, acid hydrolase and neutral protease. So both the type of granules contain three type of enzymes: specific or secondary granules and eosinophil or a primary granules. Okay. Now acid protease acts within the cell to cause destruction of bacteria in phagolysosome, while neutral protease attacks the extracellular constituents such as basement membrane, collagen, elastin, cartilage, which results in destruction of harmful tissue that is kept in check by antiprotease like alpha antitrypsin and alpha 2 macroglobulin. Okay, what does it mean? It means you can see there are two types of enzymes present in the neutrophils granules, primary granules: acid hydrolases and neutral proteases. So acid hydrolases, what they do? They do the destruction of bacteria. If you remember, in phagocytosis, the last stage, killing stage, we have included that. The after killing of the bacteria, the remaining parts are degraded by the acid hydrolases. So that acid proteases, or you can say acid hydrolases, responsible for the destruction of the bacterial products in the phagolysosome, while neutral proteases acts on the extracellular constituents. જે કોઈ બી સેલની બહારના સબસ્ટન્સીસ છે, સેલની બહારના components છે, like basement membrane, collagen, cartilage, elastin. So those substances are digested by neutral proteases. But ultimately, all the substances are responsible for the digestion or a degradation of the bacterial products. Thus, the deficiencies of these antiproteases leads to the sustained activation of leukocyte proteases, resulting in tissue destruction at the site of leukocyte accumulation. Okay, there are two antiproteases. See, alpha antitrypsin and alpha 2 macroglobulin. The name itself suggests they are antiproteases. So that's why they inhibit this protease enzyme, acid protease and neutral protease, and control their functions. Right? If their functions do not control, then what happens? They continuously damage the tissue because their nature is the destruction of bacterial products. So if bacterial products are not present, then they start to destruct the cell itself. They start to damage the tissues. Right? So these antiproteases control their functions. But there are some people too in which there is a deficiency of these proteins by genetically, by birth. So when there is a deficiency of these antiproteases, what happens? They continue, there is a sustained activation of the leukocyte proteases. This protease is acid and neutral, and they continue to destruct the tissues at the site of leukocyte accumulation. Wherever the WBCs get accumulated, they continue to damage that tissues. Okay, so that is about the granules of neutrophils. Now the granules of monocytes or macrophages. They release mediators like collagenase and elastase. So from the macrophages, or you can say monocytes, Now, there are two enzymes secreted, collagenase and elastase. And these both the enzymes are more active in chronic inflammation than acting as mediators in acute inflammation. What is that? If you remember, we have studied the two types of inflammation. Chronic is for longer time and acute is for short time. So whenever there is a severe inflammation, these enzymes are more active compared to the less severe infections. Whenever there is a less severe infections, the granules of neutrophils secretes enzymes. Okay, so these are the lysosomal components. Okay, the fourth mediator is platelet activating factor. It is a phospholipid derived mediator with inflammatory effects. This PAF, platelet activating factor, is derived from phospholipids, and it is generated from the membrane phospholipids of neutrophils, monocytes, basophils, endothelial cells, and platelets by the action of phospholipase A2. You all know that the cell membrane are consist of phospholipids. So that cell membrane of the phospholipids of cell membrane produces this platelet activating factor, and the cell membrane of the five cells, neutrophils, monocytes, basophils, endothelial cells, and platelets, releases this platelet activating factor, and that is also in presence of one enzyme, phospholipase A2. The name itself suggests, see, lipase destruct the lipids. So phospholipase destructs phospholipids, and by destructing phospholipids, they produce platelet activating factor. Apart from its platelet aggregation effect, the actions of PAF as inflammatory mediator are, that means in નામ કે છે એ પ્રમાણે platelet activating factor, that means it activates platelet, and whenever the platelets get activated, what they do, they form aggregates, platelet aggregation, and produces blood clotting. So that is the main mechanism of platelet activating factor. They produces blood clotting by activation of platelet. But along with that, what it produces in inflammation, it produces increased vascular permeability, vasodilatation in low concentration, otherwise vasoconstriction, then bronchoconstriction, then adhesion of leukocytes to endothelium, and chemotaxis. You see, all the mechanisms are already studied in vascular and cellular events. They change vascular permeability, so more fluid and cells can enter into the tissue. Vasodilatation or vasoconstriction, bronchoconstriction, then adhesion of leukocytes to endothelium. So when they are adhered, then they move into the interstitial tissue, and chemotaxis, further transfer of WBCs. So platelet activating factor is that's why one of the inflammatory mediator. Okay, the fifth mediator is nitric oxide and oxygen metabolites. Nitric oxide is a relaxation factor which is produced by endothelial cells. Nitric oxide ક્યાંથી synthesize થશે તો કે endothelial cells. It also acts as a mediator of inflammation which is produced by activated macrophages during the oxidation of arginine by the action of nitric oxide synthase enzyme. From the activated macrophages, nitric oxide can also be synthesized, and that is by the enzyme nitric oxide synthase. Synthesis કરે છે nitric oxide નું, that's why nitric oxide synthase. And they are synthesized from the arginine by oxidation process. Now what the nitric oxide do? Nitric oxide plays the two things in inflammation, that is vasodilatation and microbicidal action. What do you mean by microbicidal action? Microbicidal action is cidal means killing. So it produces the killing of microbes, the killing of bacteria, killing of fungus. So microbes can be killed by nitric oxide. So if you remember in phagocytosis, the third stage, killing or degradation stage, there were three mechanisms: oxygen dependent, oxygen independent, and third is the nitric oxide dependent mechanism. So that nitric oxide can produce the killing of microbes. The oxygen derived metabolites are released from in this same mediator we have title nitric oxide and oxygen metabolites. So nitric oxide is released from endothelial cells and it produces vasodilatation and microbicidal action. Now oxygen derived mediators. Oxygen metabolites are released by activated neutrophils and macrophages. So again phagocytic cells secreted this oxygen metabolites, and that includes superoxide radical, O2 radical, H2O2, hydrogen peroxide, and OH radical, that is hydroxy radical. So these three radicals are known as free oxygen radicals, and these oxygen radicals produce endothelial cell damage. So blood vessel wall નું જે endothelial cells છે એ damage થશે તો એ vascular permeability increase કરે. Second, activation of protease and inactivation of anti-protease. So protease enzyme જે આપણે આગળ જ જોયું, when they get activated they produce the destruction of tissue. So they get activated, so they again produces the damage to the tissue matrix and damage to other cells. They can directly produce the damage to other cells too. So there are three actions of oxygen metabolites. Now the sixth mediator is cytokines. Cytokines are the polypeptide substances which are produced by activated lymphocytes, and when the lymphocytes get activated they are known as lymphokines, and activated monocytes they are known as monokines. So cytokines secreted from lymphocytes, lymphokines secreted from monocytes, monokines. The main cytokines which acts as a mediators are interleukin 1, IL matlab interleukin 1, TNF alpha and beta. TNF નું full form છે tumor necrosis factor, tumor necrosis factor, alpha and beta, interferon gamma, and other chemokines like interleukin 8 and PF4, platelet factor 4. So cytokines is a group of mediators which include the many other chemical substances like interleukin 1, TNF alpha, TNF beta, interferon gamma, and chemokines. Okay. Now the action of cytokines are interleukin 1, TNF alpha and TNF beta produce endothelial effects in the form of leukocyte adhesion and fibroblastic proliferation. You know the leukocyte adhesion, leukocyte endothelial cells per adhere thase and then they get transferred or they can immigration, right? And fibroblastic proliferation, it is a part of the healing. આપણે સ્ટાર્ટિંગમાં જ ડેફિનેશનમાં કીધું હતું કે ઇન્ફ્લેમેટરી પ્રોસેસ જે છે that is our body's good process, કે જે ડિફેન્સ પ્રોસેસ છે પેલા ડિફેન્સથી માઇક્રોબ્સને કિલ કરશે અને પછી એ જ જગ્યાને હીલિંગ કરશે, હીલ કરશે અને ત્યાં જે ડેમેજ થયું છે એ બધું રિપેર થઈ જશે. સો એ જે ડ્યુરિંગ રિપેર જે પ્રોસેસ છે એમાં આપણી બોડીમાં એક સેલ્સ હોય ફાઇબ્રોબ્લાસ્ટ, અને એનું નામ જ છે સી ફાઇબ્રોબ્લાસ્ટ પ્રોડ્યુસીસ ફાઇબર્સ, અને જ્યાં હિલિંગ જોઈએ ને ત્યાં ફાઇબર્સ પ્રોડ્યુસ થશે તો ફાઇબર્સ એકબીજા જોડે ક્રોસ કનેક્શન એકદમ ક્રોસ લિંક્ડ થઈ અને એ જગ્યાને રિપેર કરી દેશે. સો એ ફાઇબ્રોબ્લાસ્ટ જે છે તો ફાઇબ્રોબ્લાસ્ટ મસ્ટ બી enhanced in number because of that production of fibers. So that is known as fibroblastic proliferation. Proliferation matlab that should increase in number. એનું division થાય, એક ફાઇબ્રોબ્લાસ્ટ માંથી બે, બે માંથી ચાર, like that. So that is induced by interleukin 1, TNF alpha and TNF beta. Now interferon gamma causes the activation of macrophages and neutrophils. Matlab both the phagocytic cells are activated by interferon gamma, and it also involved in synthesis of nitric oxide synthase. And a nitric oxide synthase enzyme increased high, then there is an increased production of nitric oxide. Chemokines are the substances એનું નામ જ છે chemokines. Chemokines produces chemotaxis. So chemotaxis is the attraction, chemoattractants, and so that's why they produce attraction. They are called as chemoattractants. So they are nothing but the chemoattractants for inflammatory cells. કયા cells માટે છે? તો interleukin 8 is for neutrophils, eotaxin for eosinophils, monocyte chemoattractant protein, that is MCP, for monocytes, and platelet factor 4 for all. Platelet factor 4 for all three: neutrophils, monocytes, and eosinophils. Okay, so the chemokines produces the transfer, or you can say attraction, of these WBCs at the site of injury, at the site of inflammation. Okay, so these are the first type of mediators, cell derived mediators. So summarize it again. There are how many types of cell derived mediators? 6 types of mediators. Which one? First is vasoactive amines, which includes 2 mediators, histamine and 5-HT, or you can say serotonin. Second, arachidonic acid metabolites. They also include 2 pathways, lipoxygenase pathway and cyclooxygenase pathway. Cyclooxygenase pathway produces 3 types of mediators: prostaglandins, thromboxane A2 and prostacyclin, while lipoxygenase pathway includes 2 types of mediators, 5-HETE and leukotrienes. The third mediator is lysosomal components. Lysosomal components includes also 2: the granules of neutrophils and granules of monocytes. The fourth cell derived mediator includes platelet activating factor, which is synthesized from endothelial cells. Fifth one includes 2. You can write 2 in fifth only, or you can write 5 and 6. So fifth is nitric oxide and oxygen metabolites, or you can say oxygen radicals, and sixth is cytokines. Okay, so this question can be asked in a 6 marks question. The whole explain different mediators which are synthesized from cells, or write a short note on cell derived mediators, like that. Okay, thank you.